So today I'm going to talk a little bit about climate change impact on our ecosystem. So global change in general and climate change in particular is being viewed as sort of external forces to our ecological system. So I sort of present this in comparison to a lot of complexity theory that studies things internally. So now we're going to look at the system from the outside and see how the global change impact on the ecosystem. So, okay. So to set the stage, we all know that our climate is changing. So this is from the latest report, IPCC report, showing that the temperature in the past century, we see a lot of temperature increase. So the warm color shows that temperature has increased, and this is the common phenomenon across the land and sea. So our ecosystem must have been doing something in response to that, and this is one of the big jobs of global ecologists, is really to predict how the ecosystem responds. So this is a figure showing that. So the y-axis is sort of the land flux, taking the Earth as a whole system, seeing whether or not the land is as a carbon source, sort of emitting carbon, or carbon sink, sort of absorbing carbon. And then the x-axis is year. So what we see here is that in the years that we have data, we are pretty constrained. Each line is a model in terms of the model performance. But as long as we go outside of the observations, our prediction really goes wild. So the main task of ecologists really to figure out, based on current knowledge, what is our observation and what are our experiments tell us about how the ecosystem respond. So today I'm going to talk about two case studies. One is actually very close to here. It's an experiment in California grassland. Another case study is a very different system in the eastern US forest, and see how does the grassland ecosystem and the forest ecosystem respond to global change impact. So the first one, California grassland, is really close to here. It's the Jasper Ridge Global Change Experiment right behind the Stanford campus in the Jasper Ridge Biological Preserve. So this is one of the longest experiments of global change from 98 to now, almost 20 years. It is also a multi-factor experiment, means that the experiment manipulates not only just temperature, but also things like change in rainfall, change in CO2 and nitrogen depositions. And all of them are changed either singly or in combined, so that we can have sort of a full understanding of how the system works. So this is a photo of the typical experimental design. So as many experiments of this type, the warming is implemented through the heaters above. The water is added from the sprinklers around. And then the CO2 is delivered by the tubes in here. And then the nitrogen is released by hand. So we have these four kinds of treatments. And this is the aerial view of the experiment. This is a completely randomized block design. So first we see that there are different blocks. So each polygon is a block. This is sort of the basic units of the replicates. And then you will see all these circles. So these are plot. Each one is a plot. So the CO2 and heat are implemented at the whole plot level. So there are four types of plots. We have ambient plots, we have plus CO2 only plots, and plus temperature only plots, and plus both. And then within each plot is divided into four subplots, and that's where the nitrogen and precipitation treatments gets into the plot. So again, we have ambient plot, we have plus nitrogen only, or plus precipitation only, or plus both. And then there are many replicates. At the end of the growing season, we go out and harvest the grass. Because of the Mediterranean climate, most of the grasses here are annuals. So we can use this sort of both the above ground and below ground annual biomass as a proxy for the net primary production of the ecosystem. So really the central question is, how do primary productions of the ecosystem respond to global change factors, including temperature, precipitation, CO2, and nitrogen? So if we have an experiment like this running for 20 years, we will have two sources of variations. One is that even the experiment is sitting there, we have a whole lot of natural variability, because we know that the climate in California has changed quite a bit over the 20 years. The other source of variation is that we manipulate the system. So we have the variation from the experiment. So in this plot, I'm plotting here is that the primary production on the y-axis, and then compare them with different environmental factors, temperature, precipitation, CO2, and nitrogen. And there are two kinds of symbols, and they represent from the ambient plot or from the elevated plots. So the difference between symbols will be the variation of the experimental treatment. But then the difference among the symbols, so even the same empty symbols from the ambient plot, there is a whole lot of variation. That is the variation from the natural variability. So really, what is plotting here is really to integrate both sources of variation and see how the system responds as a whole. So as we test the different models, it looks like the primary production response to temperature and precipitation is pretty much like a hump shape. So that has a better performance than the linear model. And we also see here is that across these two dimensions, the environmental manipulation pretty much span the whole entire environmental space. But the story is a little bit different from CO2 and nitrogen, where the experimental manipulation is so large, it's much greater than the natural variability. And therefore, the response is pretty much linear. So a plot like this gives us sort of a view of the system, but only marginally. What do I mean by that? If you look at any one of each panel, you know how primary production responds to temperature, but you have no idea how does that respond to other environmental factors. At the same time, we know that all the things are changing. So really, the sort of the solution to that would be to build a sort of the joint model. So here, the response variable would be the net primary production. And the predictors are the different environmental factors, temperature, precipitation, CO2, and nitrogen, and all kinds of combinations. So a couple of things that we can see from the figure is that first, the main effects are much greater than the interactions, and all the predictors are standardized so we can compare them relatively directly compare them. The other thing is that we see that the big factor from nitrogen. So if you add nitrogen, you get a boost of the primary production, as we see from the previous marginal plots. So this gives us a sort of the joint understanding of how the system works. But how do we sort of translate that to say practitioners or say rangeland managers? At least we should do something that we can translate that in the unit that they care about. So this is our attempt is to so at least taking out the results from the model and then put them into a response surface. So what is showing here are all these NPP surfaces, net primary production surfaces, and overlaying with the observations. And then this is in the 4 dimensions. So the big axis here are the temperature axis, they share the same axis. The y-axis here is the big precipitation axis. And then you will see 4 panels, and these 4 panels represent the different conditions of CO2 and nitrogen. So we have ambient versus elevated CO2, ambient versus elevated nitrogen. So we have different combinations. So a figure like this presents a lot of information. For example, if you do this exercise, sort of moving from the lower 2 panels to the upper 2 panels, if you move from the lower 2 panels to the upper 2 panels, you see an increase. And that is because the nitrogen effect, right? So you are going from the ambient nitrogen levels to elevated nitrogen levels. Likewise, if you move from the left 2 panels to the right 2 panels, you see a CO2 effect because you are moving from the ambient CO2 condition to elevated CO2 conditions. Although this effect is much smaller than the nitrogen effect, as you can see the difference. Even within the same panel, you will see that if you sort of start from the low precip, so suppose we are in ambient CO2 and ambient nitrogen condition, start from the low precip to high precip, we have sort of the hump shape, as you can see from the contour line. That is represented by the sort of the hump shape response to precipitation. And same thing with temperature, but less so than the precipitation. One of the interesting findings that we had is that we plot over the 40-year historical average in Jasper Ridge, and that happens to be here. And that point seems to be pretty close to the peak of the surface, which implies that if we have a climate change, say temperature precipitation change that is far enough deviate from the historical average, then very likely we will go downhill in this surface because this point is already pretty close to the peak in the surface, which means that if we have a sort of the climate change that is large enough in the future, we might see a decrease in net primary production. The other interesting thing that we find is that we also got the optimal precipitation that gives the maximum amount of NPP, and that happens to be at this level. And the optimal precipitations at the ambient CO2 panels are higher than the optimal precipitations at the elevated CO2 levels. And this is consistent with the physiology explanation that if we raise CO2, we will get higher rainfall use efficiency. In other words, if we have more CO2, we might not need so much precipitation to achieve the maximized productivity of the ecosystem. So in this case study, I show that the grassland production, as the sort of the indicator of the ecosystem, has a nonlinear response to climate, which are the temperature and precipitation. So we see this hump-shaped response. At the same time, the system is limited by resources. So by resources, I mean CO2 and nitrogen. So think about this, both CO2 and nitrogen are sort of the food for the plants. So if you increase these resources, you will get a higher productivity. So this kind of study shows that how the system responds to different environmental factors across different environmental treatment and also across time. So I'm going to shift the gear a little bit, talking a very different system, looking at the entire eastern U.S. forests. So the background of this is that if we zoom from the global view of climate change to a place that is more regional, for example, if we look at the eastern U.S. and make the similar figure. So what this figure shows are the observed temperature change in the 20th century. So red means that temperature has increased in the past, blue means decrease, and the size of the pixel is proportional to the magnitude of change. So we see at high latitudes, there's a lot of temperature increase. Using the same data, we can do a little bit transformation, that is getting say climate change over time, because temperature is changing over time, and divided by climate change over space. By change, really what I mean is the distribution over space, because we know that the temperatures are not uniformly distributed over space, so there's a gradient. So if we do that, we can somehow cancel out the climate and get to the space over time. So change in space over change in time, that is velocity. The concept of climate change velocity shows how fast climate is changing over space. Another way to think about this is that imagine that temperature has all these isolines over space, and over time the isoline is moving, right, because climate is changing. And the idea of climate change velocity really tells us how fast these isolines are moving. So using the same data, we can do the calculation, the climate change velocity, which shows that in the places that we see a lot of temperature increase, there's a whole lot of movement in temperature isoline, as fast as 100 kilometers per century. So that's pretty fast. Now, we got to think about this, at least in ecology or in biogeography. The distribution of vegetation, say, for example, forest, are pretty much determined by climate at the sort of a very long scale. Now, if we have a climate that is changing so fast, how does our distribution of the forest change? Are they going to do the same thing? So that's really the question. So the study of forest migration are mostly from models. So this is just an example where so this is the red line shows loblolly pine is a very important species in the eastern U.S. So the red line shows the distribution of the species now, and then the model predicts that sort of what the distribution might be in the say 100 years later. So all the models, pretty much all the models gives a similar prediction where the species are migrating very fast towards the north. That is because our climate is changing. Our climate prediction shows that the climate is moving fast. But whether or not that is happening or will happen is a question, because most of these the studies are from these models, and few of them are from observations. So really, what we are asking is, across latitudes, what do we actually observe? So to tackle this problem, let's maybe get back to a very simple question, is how do trees migrate? So we know that individual trees don't move. Forests migrate through generations. When the old trees die, seedlings grow into new trees. That's how the new range limit gets established. So really, the very simple idea here is to compare seedlings, compare juveniles, compare small guys versus adult versus the big guys, and see how different they are. If they are the same, that means we have a stable population. If they are different, that means they might have the potential to migrate. So if we have a stable population, then the range limit, we expect to see seedlings and trees have roughly the same distribution. And the northern range limit is set by the trees. When the tree dies, seedlings become new trees, and that's how that's why the range limit remains the same. If, on the other hand, we have a migrating population, then we expect to see the seedlings extend further north than the trees, right? So when the tree dies, seedlings become new trees. That's how the forests migrate. So in summary, the forest migrates through generations. Oh, okay. Yeah, so taking this simple idea, we look at the forest inventory data in the eastern US. This is a very big data set with 40,000 plots, and all these plots are fully forest, natural stands, and not plantations, with over 100 species, including the hardwoods and conifers, and over 1 million stems. And they have measurements of seedlings and adults, so that gives us the ability to compare them. Compare the geographic distributions of both seedlings and adult trees. So the first step that we did is to partition the space into different longitudinal bands. So each band of them is 1 degree in width. And then record so for example, record the northernmost seedling distribution and the northernmost tree distributions, which means that you can find the seedlings up to this point, but not one beyond. You can find the trees up to this point, but not one beyond. Another way to think about this is that if you connect all these points, that would be roughly the range limit of this particular species. Now, this is just a hypothetical example, but if this is true, that means that the seedlings are located further north than the trees. That means this hypothetical species might have the potential to migrate. So what does the actual data look like? So this is an example of American holly. It's a bird dispersed species. So on the left, what you see are the places that we can find seedlings and we can find trees. And then imagine that I partition the space into different longitudinal bands and collect all these paired values, northernmost tree, northernmost seedling, and put them on the right-hand figure. So the x-axis is the northernmost tree latitude. The y-axis is the northernmost seedling latitude. And then this is one-to-one line. If we see a lot of points above this line, means that the seedlings are located further north than the trees. In contrast, if we see a lot of points below this line, means that they are not located further north than the trees. And if they are above, then we can say that this species might have the potential to migrate. Might be a migrating species. Now, in this particular example, American holly, it happens to be that most of them above this line, meaning that they might have the potential to migrate. But not all the species are like this. We also have another example, sort of contrasting example, where by the similar way, we plot over the seedling distribution and tree distribution. So the example of common persimmon shows that by the same method, we partition the space into different bands and compare the northernmost tree latitude and northernmost seedling latitude. And in this case, it happens to be a lot of points up below this line, so which means that this particular species, common persimmon, might not have the potential to migrate. Now, one of the nice things about the analysis is that we can take a look at not only just the 1 or 2 species, we can take a look at a lot of species. And we compare about 100 species from the database. And then sort of going back to the idea of climate change velocity, this will be like our expectation, right? So if trees are perfectly following temperature change, this is what they are supposed to migrate, the speed of the migration. So compare them locally, because we know that the range shift happens at the range edge. And then put that figure like this, compare them. So the x-axis is the sort of the expected migration rate as a temperature change velocity, and the y-axis will be the comparison of seedlings and trees, the small guys and the big guys. If we have a species that is located above this one-to-one line, that means the observed migration is faster than the expected migration. In contrast, if we find the species below this line, it means that the observed migration is slower than our expectation. So over about 100 species that are fully contained in the eastern US, the majority of them are below this line, which means that many of them are migrating much slower than our expectation. So to summarize this study, it shows that for the eastern U.S. forests, the issue of forest migration, sometimes called range shift, shows that the geographic distribution change lags much behind climate change. And that actually should raise our awareness, because we already know, this is by observation, that climate is moving fast, it's changing fast. And yet our ecosystem, our forest ecosystem, they are not sort of following the pace, at least as fast as we expect them to. So with that, I show that 2 case studies on global change impacts on ecosystem. The grassland production has a non-linear response to climate and is limited by resources. Forest migration, the range shift lags much behind in climate change. So all these things shows us how the external forces, global change in general, climate change in particular, are influencing our ecosystems in different ways. I'd be happy to take questions. I don't know. It's microphone.
Microphones, yeah. So.
Hi. Thanks a lot for the presentation. Really inspiring. I wonder the differences between the expectations and the reality is maybe not only a measurement of that tree migration is not enough to recreate an ecosystem. It means that the tree itself is not an ecosystem. It depends on a bigger picture.
Right.
So it would have been depending on other species, maybe sometimes birds, even, or other things. No?
Yeah, that's perfectly true. I mean, a lot of things happens because the distribution. So this actually conveys the message, as you just said. It's not only just about the climate. There are many other things. There are soils, there are animals dispersal, and even our humans are sort of influencing the migration of the species, and that gives a lot of complexity to this problem. Yeah.
So maybe I'm not understanding it too well, but I guess I'm struggling to understand mechanically how the seedlings are so far north than the last trees you see north. Mm-hmm. Like it's miles difference. How does that occur?
You mean how does that happen?
Yeah, like mechanically, how does that happen?
Yeah. So this happens a lot in sort of in paleoclimate record. So a lot of this is by dispersal, and dispersal is one of the very important mechanisms in biology where the species, especially this plants, gets to move. So some of the species are wind dispersed. So the wind, you can think about this as a rain, seed rain. So you have a higher probability sort of to fall closer to the parental tree, but you still have sort of these long tail dispersal events that you can get very low probability, but it still happens that you have wind dispersal. Another possibility is animal dispersed. So a lot of seeds get carried by animals, and animals can go very long distances, and so that's why these kind of things happen.
One last really quick question.
Thank you. I was wondering in the graph. Okay, thanks. I was wondering in the graph where you infer migration or not migration, but when you have a lot of data points in the lower right corner, would you not be able to infer actually a range contraction?
Well, it is.
On that age, right?
Yeah. It is a range contraction. Yeah. And especially, I didn't show the result at the southern range limit. So at the southern range limit, actually, at least for some species, there is a pretty strong signal for range contraction. That's right.
Right, thanks.